So, good afternoon. Welcome to Department of Medicine Grand Rounds. I'm Ivor Douglas, based at Denver Health. Please do pick up your lunch and join us up front. It's 2 minutes past 12, and as I often remind my friend Rob Wynn, Colorado Department of Medicine Grand Round starts at 12 o'clock, so thank you very much. It's really my delight and pleasure to make a brief introduction of the Neff Lectureship, and then introduce my colleague Dave Badish, who will make an introduction of our guest speaker, Rob Wynn. This has been a remarkably pleasant 22 years of the Neff Lectureship, commemorating an extraordinary leader in our division, who I want to take just a minute to reflect on, and to thank the family who hasn't attended it for a few years, but who endowed the lectureship in the memory of my predecessor, Tom Neff at Denver Health, who was both professor here at the university on our 9th Avenue campus, as well as my predecessor as the head of the pulmonary service at Denver Health, what was then Denver General. You know, Tom had a long and very distinguished military medical career, joined as a fellow in '67, and then moved straight onto faculty where, with Marvin Schwartz and others, was highly influential in following through the work of Tom Petty, looking at the effect of supplemental oxygen, and in fact led some of the early consensus conferences around that now transformative therapy that has saved many lives. In a sad moment, Tom died all too early. He died of lymphoma in December of 1994 after a difficult but not protracted illness. And although that preceded me, my colleagues in the division, but particularly Mike Hanley, who was a close colleague, has reflected extensively both on the great gifts that Tom gave the division, as well as the excruciating experiences that he subjected his junior faculty and fellows to, known as Neffing, which was a Saturday morning intense review of the week. We're more than 2 decades into the Neff Lectureship. We've been incredibly blessed to have had some of the true leading lights of our discipline present this endowed lectureship. Nadia last year, we had a short gap during the COVID period, and this year, really delighted to have our good friend Rob Wynn, also known as King of the Peach Festivals, rejoin us here in Colorado. And Rob, maybe you'll remind them why that's relevant. I often do tell some stories about Tom, but I particularly, because of the topic of this year's lecture, want to leave you with one idea. And that Tom, although a really illustrious investigator, became a very prolific editorialist in his final years. And I wanted to just touch on a single publication that he titled The Science and Humanity of the Solitary Pulmonary Nodule. Now, for those of you who don't know what that is, I'm sure Rob will exhaust our knowledge of that in a few minutes. But in 1986, well before I think we appreciated what shared decision-making was, Tom opined in reference to a manuscript in the journal, which is now the Blue Journal, the Review of Respiratory Disease, reflecting on the honesty required to manage patients with pulmonary nodules, that in fact we manage the whole patient. And he mentioned to do this, we can only advise and educate and then support the decisions, because there are in fact too many variables, scientific and humanistic, for one best answer. And he left us with, I think, a point that really foresaw what was to come. As the facts and feelings of each patient's case change, so must we recompute our responses. And those of you who spend time in patient-centered decision-making and the shared decision-making frame will appreciate just how visionary this understanding was in an era when the patient's perspective and contribution was, I would say, secondary to the broader initiative. And with that, it's really my great pleasure to invite my colleague Dave Badish to introduce our lectureship. Thank you for your attention.
Well, thanks, Ivor, and welcome, Rob. Delighted to have you with us. So, Rob Wynn did his undergraduate work at Notre Dame, then went to University of Michigan for medical school, and from there on to Rush Presbyterian St. Luke's in Chicago to do his internship residency and chief residency. And after that, he came to join us for fellowship here at the University of Colorado. He was an outstanding pulmonary fellow and stayed on our faculty for about 12 years, I believe. He served as an attending physician at the VA Medical Center, became assistant dean of medical school admissions here, and then associate dean of admissions for the medical school, and then eventually moved on to the University of Illinois in Chicago, where he became the director of the cancer center there. And Rob is now at Virginia Commonwealth University in Richmond, and he serves as the director of the Massey Comprehensive Cancer Center there. He's also a senior associate dean for cancer innovation. And then he holds a number, it's too many to try to list here, a number of national positions. He is current president of the Association of American Cancer Institutes, and has also this year actually served as chair of the Cancer Healthcare Disparities Report. And I think we're going to hear more about that in just a moment, probably. And in addition to that, he's chair of the National Cancer Policy Forum for the National Academies of Science, Medicine, and Engineering. And so a very distinguished career for Rob after fellowship here. We're very proud of him, obviously. I've got to say that the highest compliment that I can pay you, though, Rob, is I still remember many years ago, my daughter coming home from medical school, and she said, "Dad, do you know Rob Wynn?" And I said, "Well, yes." And she said, "He's just amazing." And she absolutely loves the guy, as does everybody who meets Rob. And so it's truly a pleasure to have Rob back here to speak with us. He's going to speak on structure matters, healthcare disparities in cancer. And thanks so much for coming, Rob. We appreciate it. Welcome.
Thank you, Dr. Badish. It's good to be back home. You'll have to excuse the cough. As many of you, I'm in a room of pulmonologists, so you get this. I had a cold 6 or 7 weeks ago, around 6 weeks ago, so I have the, don't offer me any steroids, I'm good. Dr. Badish, thank you for that, and Ivor, thank you for that. I would actually want to say a couple things before I get started. There's a lot of people in this room that any success I have had has literally been mostly a reflection of the efforts that you've put and invested in me on rounds, as a pulmonary fellow, just trying to figure out how to actually do the bronchoscope and get it in the right area, to the basic sciences where I had really, I think, two major people with Dr. Nemanoff and Dr. Heasley, who I don't know if they're here today, but I would have to say that the bug of actually understanding the importance of basic science and how to drive that was really important. You had Dr. Marvin Schwartz, David Schwartz, and many other, York Miller, who all gave me that influence of how do I actually take what my love was and didn't know, passion around basic science, and translating that to the clinic. And in fact, I want to say to you that I represent CU all day, everywhere I go. And I thank you for giving me that background. I'm going to do the following, I believe, and I can do it either one of two ways. I know CME credits matter. So just for some of you, for the early birds, you can go scan this, take it right now. And I also have it at the very end for those who may come in just a little bit later. All right. These are my disclosures. And I want to set the stage a little bit about what I'm going to talk about today. This structure in the context of ZNA versus DNA and how that actually impacts our overall health outcomes. I think that most everybody recognize what Lord Calvin has stated, and we use that as the gospel. To measure is to know. If you cannot measure it, you cannot improve it. That's been something that we've accepted. By the same token, I would argue that Albert Einstein, which is, I don't know about you, but remains one of the most brightest people in the world, also said something that I think is the interplay of which I have existed in my whole entire scientific career, of understanding that not everything that counts can be counted, and not everything that can be counted counts. This interplay between understanding the basic and translational sciences and how that actually has impact within our community has been this place and space that has been evolving since the very first time I had hit a laboratory looking at MAP kinases. I've had a number of people here who, again, who have mentored me in a way, and many of you in the audience, I mean, I would list the whole crew, from my VA crew to, to everyone else around here that I actually really have to give a lot of great respect to. But I want to actually just make sure that you know that it was really, despite Marvin Schwartz's sort of call to me about why I shouldn't go to University of Washington, which I'll leave that alone, but why I should come here and involve something like there were Ku Klux Klan people out in Washington or something like that. It was a typical Marvin Schwartz thing, but it was Talmadge King who followed up and says, I understood you probably just got a call from Marvin. Let me explain. Actually, what he was trying to say is, we want you. That actually made a difference to me. We've had giants all over, from the VA to National Jewish to folks like Thomas Petty. But I have to say that between Dr. Petty and Dr. Miller, in the clinical realm of getting me to understand the importance where pulmonologists could play a role in a cancer, I want to actually have them both know, and especially Dr. Petty, that I listen. I want to tell you that it was important that this was a unique campus in which people like Andrew Kraft and Paul Bunn and Michael O'Day, which was in the field of cancer, was seamless for me as a fellow of understanding that we weren't oncology or pulmonary, we were just fighting to improve our status of lung cancer. It's that spirit that I have now I try to bring to young people that I mentor, that it really is a context of one team, one fight. Here's what we do know. Crick, Watson, and Franklin certainly made one of the major discoveries in the 1950s. In fact, most of you know that this famous photo 51 that's portrayed there was the first start of how human beings were able to start understanding the nature of the human cell. And in that understanding, we have unleashed lots of things. Phil Sharp with split genes, James Allison, which ultimately got to our checkpoint inhibitors. We've done amazing things with our technology in the context of, being able to diagnose your own cardiovascular disease or your own renal stone with your phone. We've done things like the Precision Medicine Initiative, of which I was a part of that first group in 2015 that got the first grant with our Illinois Precision Medicine Consortium, that essentially precision medicine would change the way in which we do business. We understand that we've made substantial progress in the last 20 years, but I remain steadfast in the following: that if you're Black, if you're in areas of persistent poverty, if you reside in a rural community, if you haven't obtained a college education, we know that your outcomes are worse. That issue of my love of science and trying to figure out how we could potentially get the science to do more good for people is really where this story will be framed. But before I do that, let me tell you the importance of the basic sciences. It turned out that my getting into being able to be what people now call a health disparities researcher or a health disparities expert didn't come about because I was really focused at the time directly on health equity. It was focused on trying to recognize that I wanted to learn something about cancer. We started off, as I told you, in MAP kinases and Wnt signaling, and that evolved over time into understanding this issue of stress in cancer, and understanding that while we were looking at things like DNA methylation in response, that there were other areas, RNA, and in this case, protein methylation that we did with Kamesh Bekvili. It's important to know because I want to just, again, I'm going to give you high level things and this will all fit together. Just hang with me for a second. But it turns out that the arginine methylation process was about post-translational modification of histones and non-histones. But the important part is that it played a role in many things, including DNA damage signaling and cancer development. There were several types of families. We're going to focus for the sake of argument today only on the type 1 family, which has a host of PRMTs associated with it. I'm only trying to get you to backstage because the reality was at the time we were looking at innovation and novelty. And this was, again, bare skins and sharp knives when we were starting this. We didn't have many tools, but it was fascinating to me about stress and cancer that in the background, not many people had looked at the role of PRMTs. These PRMTs, these are some of the papers that we wrote about, actually really got me interested in thinking about how PRMTs are enrolled in epithelial mesenchymal transitions and how PRMTs are involved in tumor-associated macrophages. And I just pulled up really some high-level data just to, from the papers, and they're there if you want to see them. I didn't go into all the experiments, but just to talk about our interest at the time in what was a relatively new field of looking at protein methylation and its relationship to cancer. This is an example of what we found in EMT with PRMT1, PRMT6, and the tumor-associated macrophages. We did things, as everybody would do at the bench. We had in vivo model systems in which we were able to overexpress PRMT6 and show that, yeah, indeed, the mouse models that we had grew tumors. It's interesting because these are some nude mouse experiments here where we can overexpress PRMT6, knock it out, and, as expected, PRMT6, PRMT1, they drive cancers. But that's not really going to be the story. I just want to give you a backdrop of the science. So all of a sudden, I kept thinking to myself, we know that PRMT1, PRMT4, PRMT6, all type 1 family members are involved in lung cancers. And then I kept coming across data like this. It turns out that lung cancer is still the leading cause of cancer death in the United States, and that it is also true that African-American men still to this day remain the group most negatively inflicted by this disease. The question for me was that I struggled with at that time was, how could I explain why African-American men who had the late age of smoking, fewer pack years, lower adolescent smoking prevalence, had the worst outcome? It was this issue of figuring out that could I just understand everything by the bench, or were there other things going on? And at the time, I didn't really have the language for it. But I knew that there was something else that I needed to be able to learn to be able to have impact on this problem. The factors contributing to lung cancer and health disparities were healthcare, socioeconomic status, behavioral biology. But as we start looking at genetics and epigenetics, it's interesting that most of the studies that have come out show that if you look at large-scale genomic studies, they failed to identify significant differences in somatic drivers. An example of that is the paper by Matt Meyerson and Joshua Campbell that looked at these drivers, right? Because it'd say delayed onset to cancer, fewer pack years. Why worse outcomes? Clearly, it must be genetics. Turns out that it's not so clean. By the way, when we start talking about the differences of African Americans in the context of lung cancer, I went back and actually have been thinking to myself that, well, technology is going to solve all these problems. I think most of you have been familiar also with the recent Nature Medicine paper that came out that says there was an underdiagnosis bias of artificial intelligent algorithms applied to chest radiographs in underserved populations. So all of a sudden, I had to stop for a second. I love basic science. You can blame Lynn Heasley and Rafe Neminoff for that. For some reason, I caught the bug. Look, if loving science is wrong, I definitely don't want to be right. And in this context, there was this thing that just kept bugging me. How can I explain from this pathway of stress and cancer, looking at PRMTs, how was it either the same or different in African Americans? It turns out, I'm going to skip to these slides. We're going to finish up in a minute to just talking about what I've learned at the bench, and then we're going to learn what I learned outside of the bench and how we're going to put all these pieces together. I just published a recent paper that looked at the fact that in African-American men, it turns out that both PRMT1 and PRMT6 are important, but in ways that I did not originally think about. I'll cut to the chase and say we looked at PRMT1, PRMT4, PRMT6, If you look at the TCGA databases, PRMT6 in African-American men has substantial overexpression of PRMT6 in Black men versus their Caucasian colleagues. It also turns out that ultimately, when we looked at PRMT expression levels of, again, this is just looking at Western blots and other things, that PRMT6 definitely has overexpression, is overexpressed in African-American men. Let me cut to the chase. What we found was that when we targeted one alone, we got an effect. When we targeted 6, we got an effect. But it was this interesting thing that when we were able to disrupt or target 1 and 6, we had a profound impact, and I'll get to the data in a second, on African-American men. It turns out that what we were really looking at is that 1 and 6 were interacting with one another, and that our goal then was to figure out that if we disrupted the interaction between 1 and 6, this heterocomplex, would we have an impact on cancer, or at least in vitro? And the answer was that we did. And I'm not going to show you all the data, but I'm going to just jump right to some of the PDO data that we have of collection of African-American men versus Caucasian men. What we discovered, interesting enough, was that the disruption of PRMT1 and 6 was critical, even more so to the development of cancer in African-American men with our PDO samples than our Caucasian colleagues. This is important because as we start thinking about how are we going to bring about equity, if we will, it's not only going to be just from what we do outside the bench, but I actually believe that, and I'll get back to this, that being a basic scientist, it has allowed me to look at the biology, what we call the DNA and a ZNA, differently than I had before, or if I only had one or the other. The reality is we're now developing peptides and other things to disrupt 1 and 6, and we'll keep you posted, but that's not really what I came here to talk about. I came here to talk the fact that many times I've heard my basic science colleagues, especially as a cancer center director, sort of say, well, what is my role in basic science? I have no role in health equity. I'm going to say, stop for a second and just hear me out. That typically what you're finding at the basic level, if we're really paying attention to what we believe are the other aspects of science, that is the health delivery scientists, the implementation scientists, and going into the community by definition of what I call the community-ome or community-omics, the missing omics, it will be able to help refine our scientific questions better. So I'm here to talk about health disparities. So let me tell you why I'm calling this health disparities as opposed to equity. Health equity is really a principle of an underlying commitment to reduce ultimately eliminating health disparities. Health disparities, as Braveman actually put it, is essentially the following. What actually happens when you have someone with prostate cancer? Well, when I was a medical student at Michigan, when I was a resident, and when I was a fellow, there was this belief that bottom line was if you were Black, you were going to have worse outcomes to prostate cancer. That's just the way it was. It was your biology. You had certain mutations that ultimately drove that. As Mark Twain would once say, it ain't what you don't know that gets you into trouble. It's what you know for sure that just ain't so. What we've recognized is that not only were we not probably absolutely right in the context of just because I'm Black, I would get more prostate cancer. It turns out Brent Rose and others have shown that and particularly in the VA system, with the access to the same docs, the same treatments, the same care, that Black-white difference goes away. The same thing holds true, though, for multiple myeloma with many of the studies coming out of Wisconsin. That gave me an opportunity to sort of say, well, health disparities is when you can have access to the same structures to do good, and that doesn't happen. Put another way, that yes, we have Black-white differences in prostate cancer, but it may not all be to the biology. It may be just simply due to access and other issues. So again, made up terms here. Genome, I got this from Mina Bissell when I was first thinking about this subject. Genome is to the cell as the genome impacts the individual. When I started thinking about, as an epithelial cell biologist, we used to at one point, and some of you in the room remember that we didn't think about the stroma much. It was the stroma, and really we would say that the cancer actually comes because there's disruption and deregulation within the epithelial cell itself. And the stroma, we knew that by Joan Brugge's work, Ina Bissell work, that that's not true. There's this really nuanced interplay and sophisticated going back and forth between the stroma and the individual epithelial cell that results in accumulation of issues that then result in cancer. I took that model and started thinking about the stroma as being our environment, if you will, the genome, for those made up word, but for those who are comfortable with it, just to make this genome, all that stuff within the exosome and everything else, and the interplay it actually has with an individual. As I started thinking about this, I had to go back in time to actually look at Dr. Broder, who was the NCI Cancer Center director in 1989, who actually also argued that poverty was also a carcinogen. So how do I make these two worlds go? That I know what happens in the basic science world, but the one little peek that I actually had beyond the bench, I understand that there was an additional world that was probably also having an impact on not only outcomes, but biology. I wanted to show you this because in a context of structure, I show you this because it was interesting to me. This is a happenstance, by the way. I was going through some historical books and some health issues and found out that these were the areas that had the most hardcore Jim Crow laws as they were applied. And for those of you who don't know Jim Crow, that was been a while ago, right? But when I look at where the the areas that have the lowest life expectancy and the areas that had the worst outcomes from cancer, it's interesting how things continue to track. That gave me an idea. It started getting me in this mindset of thinking about that structure and place and space maybe really does matter. And again, let's take a look at this at the local level. This is where this is where I had an aha moment. That actually ZNA, i.e., and I see a couple people in the audience like, what the hell is ZNA? Your zip code neighborhood of association. And its relationship to the DNA was a space of research that needed to be looked at more robustly. How does this work? Let's take Chicago for an example. The life expectancy in Chicago, as you can see to your right, is 85% if you're on what's called the loop. If you're confused about Chicago, that's where the Magnificent Mile, let me say Magnificent Mile, but beyond the Magnificent Mile, if Oprah is going to go back to Chicago, she's going to the loop. If you literally go 4 miles west, the life expectancy drops by 16 years. Not shown here, but if you go 8 miles south to what's called an area called Inglewood, the life expectancy drops to 30, by 30 years. Now, initially when Steve Wolf came up with these things, we were thinking, it's probably related to gun violence because people know Chicago's a rough place. Believe me, I was there. Turns out that the gun violence and all the rest of that stuff was negligible. So as you start thinking about why would you have 30 years loss of life expectancy 8 miles south of the loop? One had to say, is it just because the people in that area are just biologically disadvantaged? It turns out that this is where I started putting the pieces together that could I get information from the outside, meaning beyond my bench, that can help me refine what my scientific questions were at the bench and put it all together better. Redlining, which most of us know, happened throughout the United States, based on Dr. Homer Hoyt, who is the head of the National Housing Act then in the 1930s, actually set up this thing based on the soft science, not soft science, it was based on the best science of the time, called eugenics. As a result of that, they divided up who they thought was smart enough to would be able and capable enough to pay home loans. It turns out Didn't have to tell you anything, but that there was a difference between blacks versus white. So let's just take this for a second. This is Richmond, circa 1935. And what you're seeing to your right of the map is that the areas are called undesirable areas. That's called the East End. Let's just call it that way. And the West End, which is the blue and the green, is considered to be the desirable areas. Well, it turns out that while redlining, we still have some modern day redlining, it made some difference, but it was really this issue, which I did not find out about until later, of the urban renewal program, i.e. building white roads through black bedrooms, that you saw all over the country. Why does that matter, you're saying, Dr. Wynn? Well, because I grew up as a kid thinking that as an African-American, I just simply was more predisposed to having worse outcomes with cancer, not really recognizing that, for example, that not only were those highways in areas where you had areas of persistent poverty or areas of color getting diesel fumes and all the rest of that, but the reality is there were now businesses being put in those areas that we didn't account for. Let me put it a different way. That work done by Vicky Seawald and Tessem and others, and this is new work, and the newer work by Dr. Seawald, actually is really defining for us that particulate matter 2.5, which is associated with increased risks and poor outcomes of cancer, are disproportionately and systemically more concentrated in areas where you have communities of color. So this concept that, as an African-American, we talk in this rough terms, African-Americans are more predisposed to cancer because of their biology. What if it's not only their DNA, but what's going on in the ZNA, that zip code, the neighborhood of association, and the intersectionality of that on our biology and our outcomes. Epigenetics, by the way, is more than just DNA, by the way. We have RNA, we have protein, so that field is also getting to be better. So here's another way of looking at it. I'm going to get into one of the grants I just got. We're using, and I won't get into all the details, but we're using machine learning and lots of other AI systems to sort of put some of these things together about what we call multi-level stress clusters and defining these multi-level stress clusters and how they are actually impacting cancer. For example, you all know that where there is lesser tree canopies, well, let me put it this way. Poorer communities live in areas that typically have higher surface area temperatures, literally because the tree canopies and the density of those are usually less. So I've been going to Baltimore and areas like that and looking at the surface area temperature differences in the lack of tree canopy and all these other things as we're now getting hipper to this of how that actually is contributing to chronic diseases and chronic diseases like cancer. We're starting to actually use machine learning, and I'll go into this later, but machine learning and some other things about helping us to identify when a patient comes in, could we actually understand not that they're Black, but that the place in space that they come from, i.e., that if we could calculate the multi-level stress clusters, will we have a better indication of their risks? And to better inform us about the early interventions and interceptions that we should come about. By the way, we're using African Americans, but let me just pause for a second and say that if you had the choice to be a 70-year-old white guy, and they said live in Kentucky, we'll pick one, Beattyville, Kentucky, or to live in Utah, pick something, Salt Lake, you would be wise to pick Utah. It turns out that if you're a white male, living in an area like Kentucky, you have 3 times more likelihood to have a poor outcome from your cancers, particularly lung, but there are others. So we need to stop playing and expand our minds in the context that it's not just someone's walking into your room and you see something, whether it's Latino or you see someone that's African-American, it's their place in space. The missing omics. While we have lipidomics, genomics, proteomics, there's a whole bunch of omics. But typically when you're sitting in front of that patient, you're missing their universe, that community-ome, that the data from that can really help inform you about what we should be thinking about. The interesting thing about that is that I'm gonna just take a couple minutes to go through these slides because this was fun. This is an R01. We got this R01. This was kind of one of the fun R01s because you could just think out of the box and just say, well, what? Come up with something. Literally come up with something. So we did. We called this Recode. I do this with Sage Kim at University of Illinois. So here's what we looked at. what would we think about, how does where people live affect exposure to risk, particularly cancer risks? And actually, as we started looking at communities, we started looking at life experiences, et cetera, and how that would have an impact on our outcome. So let me get to the good stuff. So here's what we did. Instead of looking at black, white, we literally, in Chicago, there are 77 communities, but 50 wards. We looked at black men in all 50 wards and just simply asked the question, rank them from highest to worst outcomes in their cancer outcomes, and particularly lung cancer. So we did that. We also did something kind of interesting. Now, I didn't look at ADI measures or any of that stuff. We just simply looked at homicide rates to make it simple. Now, was this something that we knew was going to work? I did not. This was something I was like, let's try it. So we literally just said ADI, all these other metrics are straight. Let's just homicide rates. It turns out from all of the 50 wards that when you looked at the areas that were the most distressed and we looked at their collective cortisol, the cortisol levels were higher. That makes sense. Okay. Association. But let me get to the fun part. So African-American communities that actually were more stressed, associated with more gun violence, right? And all the rest of the stuff that kind of goes along. But we just said gun homicide. As a collective, we went into the barbershops, gained trust and all the rest of the stuff. Cortisol, right, was higher. But this was the most interesting thing for me. We started getting deeper and started asking questions about, could we get genetic samplings? And so again, we went to all of the 50 wards in Chicago, got relatively decent samples that we could get. And what we showed is that those areas that were most distressed, interesting enough, had gene expression changes of disruption in DNA repair, cell cycle, and many other assets. Now, the funny part was we then said, okay, that's kind of interesting, but what would actually happen if you went to the tumors? So again, looking at all 50 wards, which you had reasonable data for, again, we looked and you can see that in those patients that came from those areas with cancers, by the way, that we saw a different pattern, molecular pattern, of those people in the more distressed areas than we did in just say an African-American coming from an affluent area with a median income that was above 100. So this issue of just saying that Blacks, which by the way is a social construct, we need to be able to refine our language. It's not so much, yes, that biology does matter. And there are definitely some mutations that may be unique to certain populations. But I remember and remind people that there's something called ancestry. It's not just African Americans with ancestry. We all have ancestry. How does this fit? There's work been done by Melissa Davis and Lisa Newman that said when you talk about African American women with triple negative breast cancers, you just can't say African American women because those with more Eastern African ancestry have a much better outcome of their cancers than someone with more Western African ancestry. So as we talk in this very crude terms about Black versus white, I'm reminding people that even when you say white in areas of persistent poverty versus, white folk or a white person that may live in an area where the median of income is higher, someone with more Eastern European ancestry versus someone with more. I think that the funny part is I started off by sort of saying that the benefit of precision medicine was that we were going to be more precise in our language. I think we're being called now in the 21st century with the data to be more precise in how we're looking at the patient that's in front of us. I'll end with sort of saying real quick that we have really talked about, Harold P. Freeman, this issue about discovery versus the disconnect between delivery sciences. I actually also think that there is a disconnect between the delivery and the time of diffusion. I'm writing a paper right now, again, and Jamie knows this is quite painful. You hate when people, like at these journals, they call you up and they say, yeah, we want you to write a paper. Then you're like, well, I'm on my damn second revision. How does that happen? Right? But anyways, the paper is going to be called, the title, we're writing for Nature, Innovation Does Not Equal Impact for All. And as any good pulmonologist would do, I had to come up with an equation. So I just borrowed a fixed equation for diffusion gradients. And interesting enough, the funny part about this is we're starting to modify interesting of Fick's first law, and we're actually using that, and now we're going to make it almost I don't know what to call it, but we're going to call it the social Fick's equation or something of the equivalent that says the following, that once we actually have discovery, the discovery sciences comes from a basic, that basic science then translates. What traditionally happens is it gets translated to a small slice within our communities. That small slice typically have resources, have access. And so for me, it becomes this issue of the diffusion gradient. It's not in some cases that we don't see that the best of our technologies and the best of our science doesn't eventually get to everyone, but how long does it take? And what are the obstacles in its way that will allow for a better diffusion gradient with some and a worse for others? You see, this new science has actually gotten me thinking about the fact that, and part of the paper is that with every new innovation, it does not always equal impact for all equally. In fact, even if you go back to mammography screening, or hell, let's just go back to COVID. When we actually had the mRNA vaccines coming out, there was this black-white gap that had to be closed because we became more aware of it. But again, I say all of this to you because I think we're at a new place in science where we're not only having to understand the basic translational, but the translation of that to the impact within our community, as I'm putting this collective diffusion science. In that context, I said that we've been getting in the way, not only of, is it the social drivers of health, not only is it pointing the behavior issues of the people living in the community, but the issue is, as health systems and academic systems, what have we also contributed to the unevenness of this diffusion? By the way, this time for we should be retiring the hard and soft science conversation. That's ridiculous at this point. In fact, you know what? I will not even put up with anybody in the same room talking about hard and soft science anymore, because here's a fact. When we came up with the human genome, what was hard about that? That was just sequencing. The hard sciences came as a result of that finding. And by the way, when most of us are now interested in data sciences, the hard sciences of AI, you're measuring freaking behaviors, which by definition is a soft science. I think we ought to start figuring out more about how to align the sciences as we're thinking about innovation and impact. And I really believe this frameworking of impact allows us now to think about how to align our basic sciences in a way where we can bring most of this community omics to you all as basic scientists to help you refine your scientific questions, and how we can have better discussions between the basic translational sciences and our community implementation of deliveries so that we're aligning our efforts for the ultimate outcome. I'll stop with this last slide and say that one of the things that, again, if people talk about, what I've learned from CU, it's from people like Dr. Petty, Dr. Badish, all you guys, Dr. Schwartz, Dr. Marvin, David, I think he's here. So to just not take no for an answer. So I adopted something that I got from a guy I learned, I think it's my Angelo, but I got it from John DeMonte, who says, when you're thinking about changing a system, it's not enough to do something different, do a different thing. So part of the different things that we actually have now, the only embarrassing thing is the name. I don't know why the hell they called it this name, but just blank out the name and let's just talk about the program for a second. This was a way of how do we get more people on the clinical trials? It turns out that we were training people who were really good in designing and implementing trials, but had no idea about the science of outreach and engagement. By the way, we have a whole crew of people who were doing outreach and engagement, but had no idea how difficult it was to get a molecule to become a medicine. So this program, which now has graduated its first class, was a dream of saying, what if you broke down the barriers and you taught people who actually knew how to design and implement trials, how to use an asset map, how to actually understand the tool sets of the frameworks that are part of the science of outreach and engagement? What could that do? And in fact, I would tell that the first pancake was not bad. It turns out now I have a handful of people who not only are they writing their protocols with the various arms, but at every moment they're writing down how they could get rural populations or a Latino population onto their trials by doing X at this step, X at this step, Y at this step. It's beautiful. I think hopefully it'll be game changing. I'll stop there and just say that I'm super happy to be here. I wouldn't have been there without you all. I am probably the most proud I've ever been about CU. In fact, probably more people hear about CU outside than since I've left than they probably want to care to hear about. It's not to say that U of I hasn't been important, that VCU hasn't been important, but this is my home. This is the place where it all came together. This is the place where you taught me and inspired me to do a different thing. And for you all, my friends, my colleagues, my mentors, I just want to say thanks. And hopefully, as always, from our primary care folks like Mark Ernest, who's, I don't know if he's here or not, or all the rest of these guys, that all the pieces have come together for me to be hopefully a good reflection on CU. I'll stop and just say thank you. I hope I've never embarrassed you, because the truth of the matter is that I couldn't be where I am without the start and the foundation that I got from this place. And I'll always be above 24 ways till tomorrow. And I appreciate you. Thank you so much.
Rob, as I said earlier, we're so proud of you as one of our fellow graduates, and thank you for everything that you're doing. It's wonderful. That was a great talk. You have an unique ability, I think, to look at the basic science underlying risk for development of cancer and how that intersects with racial and socioeconomic factors impacting risk. It's a unique background that you have, and a wonderful talk. Thank you. I was going to ask you if you believe that the effectiveness of screening programs depends upon the risk in the population that's being screened. It seems like these socioeconomic and racial factors ought to be taken into account when we decide like where to target our screening, where to focus it and whatnot. Are we doing that? I know you're active on a national policy level right now. Do we target screening to a sufficient extent to those populations at greatest risk for developing cancer?
You know, it's interesting. We talk about screening, and it's an interesting place right now. It really is. Jamie and I have some are definitely front row seats to figuring out how the screening efforts are going to come about. But here's one of the things I'll tell you. I was just talking with some folks at NPR about the better numbers happening, for example, African Americans in the context of lung cancer. And it turns out, you know what most of this was related to? It was related to reduced levels of tobacco use, which is no surprise because there's many other alternatives these days, and access to actually treatment. When we look at our screening rates, we should be doing better. And the wonderful part is I still am a rabid fan that screening does work, but Otis Brawley would be the one that sort of argues that too much screening can actually be bad. So I've actually pushed back on Otis and said, in an era of precision medicine, in an era of person-based medicine, if we had more data that would actually help us to figure out who we should target or which areas we should target, we might be better off. I'll give you an example. We did this when I was in Illinois. I played with this sort of approach where we looked at the wards, and instead of looking at zip codes, I looked at political districts. Now, initially, people said, what the hell are you looking at political districts for? Just look at zip codes. Well, the problem is with zip codes, for example, as we start talking about smart screening, is that zip codes are great for mailmen and great for academics. But beyond that, they're not really all that useful. Interesting enough, when I was able to break it up into not only wards, which we could do by, delegates and state reps and all this other stuff, but let's just take it on a small level. We were able to, for example, show in Inglewood, that area with 30 years life expectancy decrease, that there were 3 times more people in the 20th ward dying from prostate cancer than the rest of Illinois and the rest of the United States. It turns out that by that having that kind of data, we were able to have better conversations with the mayor then, who was Mayor Rahm Emanuel, and the health departments to figure out what could we do in Inglewood to change the tide. Long story short, we were able to get resources and work with not only the FQHC networks, but the neighboring hospitals within that area, some of the community health to change that tide. So I think screening is actually key. I disagree a little bit with Otis. Otis Brawley, I mean, he's a wonderful, he's again a mentor, and I love that guy. But there are some areas where I still think that screening, until we can actually really get to the magic bullet where we can pull something out of, your sputum or something that says you're absolute, screening is important, and we still should be pushing it. I would argue that in our rural communities, we're underutilizing it. And in our areas of urban underserved areas, at risk, what we call population Latino African Americans, we could certainly do better. But if you look at the numbers of what's happening right now with cancer and some of these chronic diseases in the rural areas, we suck. I mean, the numbers are actually getting worse year by year. So the crazy disconnect is we have all this technology. We have all these promises of what science is going to do, and yet we have areas where that diffusion is not there. And so they're not benefiting. And so that's going to be one of my big goals, and definitely working with the National Cancer Policy Forum to really start thinking about policies where we can really have more people benefit from our science.
Are there questions from the audience? David?
And I know that was a lot to fit into one talk about the basic and all this stuff, but hopefully it hung together.
Pretty good. Welcome back, Rob. Good to see you. So, one of the things that your talk really speaks to, and I think one of the many lessons that we got from taking care of patients with COVID for the several years that COVID existed in the United States was that we don't have a healthcare system and we don't have a public healthcare system, and that countries that had a healthcare system or have a healthcare system and had a public healthcare system did much better with COVID than we did. And so the question that I have is really looking for solutions, looking for how do you address this disparity issue in more of a population-based way? And to what extent have healthcare systems or public healthcare systems done to address this issue in other countries? And are other countries that have these systems in place, are they doing better in terms of the disparity of healthcare and the diffusion of healthcare in the population?
Yeah, that's a great question. It's good to see you. So what do we do? We recognize the problems. What do we do? So part of my pitch has always been that we are underutilizing the tools that we have really at our access. As health systems, I'm going to pick on cancer, for example. We have these comprehensive cancer centers, but most of the cancer care that gets actually done is outside of our systems. We don't really have an alignment and a partnership with the ACCC in a way that I think we could have in a context of bringing things together. So one of the things we've actually experimented with is that Massey actually has a network of what I call a part of the National Community Oncology Research Programs of getting, for example, trials out to these areas, rural areas, underserved areas. That has really been an actual helpful of the alignment of those things, as well as better use and impact with federally qualified health centers. We usually, federally qualified, actually, the one thing I learned in Chicago was that, we treat FQHCs as if they were a 1950 car from Havana. We should be rethinking about, that's the undergirding of our system. How can we rethink and reimagine what could actually happen within those FQHC spaces that aligns with, sort of our academic sort of centers? And I think the experiment was done actually in Chicago. And what I found out was that the benefits were that we could, within not immediately, but within a 3 to 4 year, 5 year period, or at least a 3 to 4 year period, start seeing some metrics that were actually starting to change. So I think it's not as if we don't have the things. We just, what is that old thing? We're not so much dysfunctional as we are disconnected. There are places that we have that are relatively disconnected. And by the way, the CMS, which we fought hard for and finally got some victory of having CMS reimbursed for navigation, that you're not going to convince me that one of the elements of this is that we don't really understand navigation at its fullest in the context of community activators, and those community activators and the trust that they build with our community health navigators to get people to the care. So I think that there are tools that we're going to be actually looking at. Navigation is one of them, better use and alignment with community centers and certainly within our FQHC spaces. But it's a great question because there are a lot more pieces than just that. But those three, I found out over time, just aligning those a little bit better could have at least more impact. I don't know about the impact we want, but at least better.
Professor Nguyen, I don't have any questions, just a couple of comments. First, the title of the Diversity in Clinical Training Program is correct. Well-deserved, and thank you for your leadership on that. The second one is I've already have the email of the hospital director at Fleming County Hospital in Beattyville, and I will be working with them on their screening program as soon as possible. And so we can help you eliminate the reference to Beattyville, Kentucky in your talk.
Thank you.
Rob, great to have you back. Thanks for you imply that the interaction of two epigenetic regulators enhances risk in Black males compared to other groups, suggesting an epigenetic effect, perhaps of environmentally modifying gene expression. We also know that epigenetic marks can be intergenerationally transmitted, and we accept that there are many disease states and trauma environments where transgenerational transmission has been shown to be important. Do you have any suggestion that that may be applied in this environment either?
Yeah, just because of the sake of time and all the rest of that stuff, we didn't get into that whole thing about imprinting. You know, we've gotten data all the way back from World War II about the concept of imprinting, right? And one of the best examples in African-American women, when we talk about the high infant mortality and, you know, rates and all the rest of the stuff about that, Beyonce and Serena Williams should never have had any problems. And why are they having problems with, you know, the infant mortality type deals? This is an interesting concept about that whole thing about under your skin, getting under your skin and that principle. We are starting to, I think we suffered from not having the tools to be able to unpack some of these things now. By bringing some of the social scientists and our basic sciences together and having different kinds of conversations, we're starting to develop different approaches to be able to figure that out about what portion of this is the dose response in the context of imprinting and what part of this is just the acute situation. It's hard. But, you know, everyone keeps telling about the social sciences. Oh, my God, it's just so hard. But it took you freaking 125 years to get a freaking checkpoint inhibitor. Come on, quit playing with me. Right? From the time of Cooley to the time of checkpoint inhibitors. And by the way, we were there when we were in the front row seat where we saw some of the clinical trials that actually were not great. And yet we stuck with it because we wanted to try to solve the bigger problem. So for me, that's a great question. And I have now recruited two other people that are much brighter than I am, we're starting to put together our heads about how to unpack and come up with ways to address that issue about passing things on to generation and what that means and what it looks like. There has been some groundwork already laid in that area. So it's not as if we're starting from scratch, but I have two bright people that are actually starting to really put those things together. So I'll keep you posted.
Right. Well, any other questions? If not, Rob, thanks again for coming. Terrific talk and great questions too. Thank you. Take care.